happy to introduce our special seminar speaker today, Professor Fai Au from University of Iowa. So Fai had a really long history of training in different fields. So he got his Ph.D. in structural biology from Oxford University. And then he went to Stanford to work with Professor Wing Wang in the Department of Statistics, where he first started working on computational methods for analyzing Illumina RNA sequencing data. And at that time, he developed one of the earliest short-read aligner called splicing for identifying splice junctions from RNA sequences. And then with the technology moving in terms of high-throughput sequencing, he then went on to work on methods for analyzing long-read sequencing data, in particular long-read RNA sequencing data generated by Fai Au sequencers. And he has published a few papers on tools for analyzing long-read sequencing data, and also a very seminal study in DNS a few years ago where he combined the long-read sequencing and short-read sequencing for comprehensive identification of novel gene isoforms in embryonic stem cells. And one of the important implications from his study was that even for very well annotated transcriptomes such as the human embryonic stem cells, there are still a lot of novel isoforms to be discovered once you combine the long-read and short-read technology. I think this is what he referred to as the hybrid-seq technology, where you use both long-read and short-read for transcript identification and quantitation. And then he took a faculty position at the Department of Internal Medicine in the University of Iowa, where he actually took my old office when I moved out of Iowa. So where he continued working on projects where he really expanded his research directions and used those hybrid sequencing technology for a variety of interesting applications, including isoform discovery, detection of fusion genes from cancer transcriptome data, and so on. And he has received a number of awards, including a Young Investigator Award from the RMA Foundation this year. So he's here as part of this summer program in computational genomics, and we thought it would be great for him to give a 1-hour presentation about his research, and as I think there will be a lot of interest about this work at UCLA. So without further ado, I'll pass it on to Fai.
Well, thank you very much, Yi. Thank you very much for having me here today as well. Well, today I would like to talk about how we make use of the hybrid sequencing to analyze transcriptomes. Well, the first part, well, actually, I think Yi has made a very good introduction of hybrid seq, but I think I also want to tell you why we want to use the hybrid sequencing to analyze the transcriptomes. And the second part of my talk would be the methodology development, how we make use of the strands of 2-generation sequence data to solve a couple problems. For example, error collection by LSC, isoform detection and predictions by IDP, and how we analyze the fusion gene using the hybrid sequencing data, and also doing haplotypings, and also the allele-specific expression quantification using the hybrid sequencing. Well, at first, I would like to talk about what is hybrid seq and why we want to introduce the terms hybrid seq in the transcriptome analysis. At first, I want to give you an example of the importance of the gene isoform expressions. Okay, in 2011, a paper published in Cell shows that there's 2 isoforms of this gene called FOXP1s. Well, these 2 isoforms have the only difference in the usage of 2 exons. The typical isoform use the blue exon and skip the red exon and have this specific domain targeting to the this motif, and this motif enriched in the differentiate genes, so it regulates the differentiation process. Well, but they find a novel isoform from these genes that skip the original exon here and use the novel exon, the red one here, and have a novel domain targeting to this motif, and this motif enriched in the polypotency genes. You can see that a single gene could have completely function. While one can maintain the polypotency, the other one can regulate the differentiation process. So you can see, well, if you want to understand a gene function deeply, thoroughly, you have better to understand which isoform it expresses. Okay, well, here is the isoform length distribution in the human genomes. We have peaks around 2,500 base pairs, and we also have a significant numbers of genes in isoform longer than 5,000 base pair. And a lot of method has been developed to use the second generation sequencing, I mean the Illumina sequencing data, for example, from 50 base pair to about 300 base pair to reconstruct the isoform, or so-called transcript. However, it's very difficult. Although many methods has been published. So for example, coupling, I think the citation is a few thousand now, so it's very famous ones. It used the coverage model, and he make a graph using the junctions between the exon, and then it outputs the polynomial set of the gene model, gene isoform, that explains all the paths from the 5' end to the 3' end, okay? But the problem is why it is polynomials. Well, no biological evidence say, well, cells or a transcriptome must express a polynomial set of isoform, okay? This is a mathematical theory. It's called the Worth theorems, that in this given graph, we can always find a unit and polynomial set pass from one end to the other end. But this is mathematical. But what we are working on is a biological problems. So when you apply some non-biological constraint to a biological problem, you always have a lot of problems. Well, although coupling is very famous, I think many people, when you analyze the RNA-seq data, your collaborators will ask you to run coupling because yes, it's most used. But most used does not mean reliability, okay? It reminds me something happens in 2012, okay, when I planned a trip from San Francisco to Beijing. I typed San Francisco, Beijing in Google Map, okay? If we count the citation of Google Map, we may have tens of thousands, or even millions of citations of Google Map, okay? However, it gives me this solution. Well, I have to go to Seattle first, and then Hawaii, Japan, Shanghai. I think this is definitely not some things we are looking for, okay? Well, although it's very famous, most used, but it does not mean it gives you a reliable results. Well, but the problem comes to us is whether we can get a reliable identification of the gene isoform from the short-read data, okay? Well, this is some example I give you, but actually theoretically in 2012, a couple papers has been published to show that, well, if you only have the RNA-seq short-read, it's very difficult to reconstruct the gene isoform. Well, I highlight two keywords here. One is short, one is MBR. Well, we cannot change MBR here, but we cannot change this, short. That means if we have longer reads, whether we can have a better reconstruction of the gene isoform. Okay, well, come to 2016, well, I think maybe some programmers in Google, well, went to my talk previously, they find this bug, and then they find some other information, and put this information in Google database, and then they find, yes, we can take the air flight, okay, from San Francisco to Beijing now. Okay, that's something like long read. Long read is something can link something separated by a long distance, right? So if we have some information in our data, well, then we have a better solution now. Well, yes, there's a good news that third generation sequencing, for example, Hepa sequencing, Oxford nanopore sequencing has been developed for a couple of years. They give us a few thousand base pairs and up to about 20,000 or 30,000 base pair, okay? But they give us a very long reads, but the average is pretty high. It's about 13%, okay? Well, compared to the about 0.1 to 1% average in the second generation sequencing, this is a very high average. So we have to find a way to overcome this problem before we make use of the long reads. Well, take the Hepa sequencing as an example. Well, they have a very clever way to do the error collection by the data itself. Well, see, this is the small chips for Hepa sequence. Well, in these chips, we have about 150,000 small holes called ZMW hole. And in each hole, we have a polymerase, which bound to the targeted DNA for the sequencing. Well, this is the targeted double-stranded DNA. Okay, if you have an RNA, you can convert to the cDNA, and it add some artificial sequence at two ends so that we can generate a circular DNA. And a DNA polymerase can bound to a motif in this artificial loops and then start replication. During the process of replication, we can capture the signal, and then we know what the sequence is. Well, the raw data is called continuous long-read CLR. And the error rate of this raw data is 13%, okay? And there's a very clever way to do the self-error collection from the raw data. Because we know the sequence of these adapters, we can trim this adapter off. Okay? And then what remain would be this so-called subreads, okay? This is just a copy of sequence of the same molecule. Then they can generate the consensus sequence, so-called the CCSV. Well, after you generate the consensus sequence from this raw data, the error rate can come down to about 5% or even 1%. It depends on how many subreads you would have. Well, this is a very good strategy. In fact, my Snyder's group in Stanford has been using this strategy to analyze a lot of transcriptomes and have a good publication a couple years ago. However, well, this strategy will sacrifice some things. It will sacrifice the read length, or you have a better, higher accuracy of your data, okay? Because if you do a simple math, then you will find why there's a trade-off between the accuracy and the read length. Well, because if you want to have a high accuracy of your CCS read, that means the more subread you need from a single molecule, right? Well, and the numbers of subread from a single molecule depends on two things. One is the total length of CLR, total length, and the other factor is the final read length, the target molecule length. However, we know is that this one, the total length of CLR, depends on the lifetime of the polymerase. If the polymerase dies, the replication process stops, and this is what you have. So if this is a limited number, and then if you want more subread, that means the shorter CCS read you will have. Okay? So this is a trade-off of accuracy and read length. So we have a lot of high accuracy short read already. Okay? This is not what we really lack. What we want is something like the length information, because if you have the length information, you can link the exon together so that you can find the isoform. You can link genes so that you can find gene fusion events. You can do the haplotypings by linking the SNP events. Well, but we never forgot the problem of the high error rate and low throughput of the third generation sequencing. So that's why we introduced a strategy called hybrid sequencing, hybrid 6. Well, to integrate the accuracy and throughput strengths from the second generation sequencing and use the length information from the third generation sequencing. This is called the hybrid 6, okay? Well, to make use of the hybrid 6, we have to develop a set of bioinformatics tools to integrate them together. The first step is the error collection, okay? We use the short read to correct the long read so that we have the error collected long read. The second step, to make use of the short read and corrected long read together, we identify gene isoform and quantify the gene isoform. In the third step, we have IDP-ASE. We solve the haplotype. We do the haplotypings with the short read and long read, and also we quantify the allele-specific expression at the gene level and also at the isoform level. And at last, we detect fusion gene events and determine the fusion site at a single nucleotide resolution, and also identify and quantify the isoform expression from the fusion genes. Okay, this is the pipeline we have so far. Okay, first step was error collection. Well, we developed a tool LSC for the error collection. We use the short read to correct the long read. Well, it's kind of similar to as playing a puzzle, okay? Well, when you have by a puzzle, well, you always have a pictures on the top of the puzzle. This is what you really want to make, okay? But the resolution is low. It gives you the global information, but the resolution is low. But when you open the box, well, you will find some high resolution small p6 things. This is something like what we have is short reads, right? What you want to make is a big pictures, high resolution picture, okay? Well, this is something very similar to our error collections process. But what is the first step of playing this puzzle? I know some people from the CGSI already heard this process before, but how about the strategy we have if you did not hear this before? What's the first step when you play a puzzle?
Border.
Yeah, border. And the other strategy? No one want to try? Okay, things. Huh?
Something in the special color.
Yeah. Well, see, I pick up this one. This is a blue one, okay? I won't put it to the lower left corners, definitely not, because we know this should be greens, right? We will put it to the upper right corners because these blue ones likely come from the sky or the lake, okay? Well, this is very similar to a process in the sequence analysis. alignment. Okay, alignment is to find where something comes from, right? So actually, the first step we do in the error correction is to align the short way to the long way. Okay, well, if we have, well, but the alignment process have a very fundamental assumption that, well, the target sequence should be very similar to the reference sequence, right? Otherwise, you cannot align to them, okay? However, we have a high quality short way, low quality long way. They are not very similar enough, okay? They are, so the high error rate of the long way prevent you from doing the alignment of the short way to the long way. Now it becomes a chicken or the egg problems, because you want to solve the problem of the high error rate, but the high error rate prevent you from doing that. Well, so we stop here. Well, this is the end of my slide there. Okay, but we find a tricky way to overcome these problems. We know that a lot of errors in the Hepa data come from the homopolymer runs. What does it mean? Well, if the true sequence is 6A, well, the Hepa sequencer can give you 4A or 7A. That means, if you give you 4A, you have about 2 mismatch, right? And the distance, the editing distance from 7A to 6A is 1, okay? And now, if we can compress this homopolymer run to a single A, then the editing distance between them is 0 now. That means you do not consider any mismatch between them. So actually, well, in this example, we compress this G to single G, this 2G to a single G, and we do not have this mismatch anymore. And we can align the compressed short way to the compressed long way with high sensitivity with a very short running time. Okay, well, when we do this compression, we can do the alignment. And what else we do? Well, actually, it is very simple. Well, we do the compression first and then apply any short way aligner. For example, bow tie to BWA, then you can align all the short way to the long way, and this is the result you have. What you need to do is to compare the difference between the short way and long way. In this alignment, the aligner could tell you some point mismatch, insertion, deletion, and you also need to consider the difference in the compression, the homopolymer compression point, because originally, well, the homopolymer runs in the long way is different from the homopolymer in the short way. Well, overall, we replace the long way sequence by the long way sequence by the short way sequence, and then we can finish the error corrections process.
So how much of the error comes from this homopolymer?
Good questions. Actually, I mentioned that the error rate of the Hepa sequencing data is about 13%, right? About 6 to 7% is from the homopolymer run. That means about half of the error rates come from that. Most of them is the insertion deletion. Okay. Well, and before and after error correction, this is something we have when we publish the paper, but actually the performance is much better now. But in these original versions, we find that, well, the raw data have a peak of about 85% because the error rate is about 15%. And after error correction, most of them moved about 90%. So you have a much better error accuracy after error corrections. And
so are those like homopolymer errors random, or do they always occur on the same position? You know, if you look at different reads of the same gene.
Well, actually, we did not study that, but papers we are working on is to study the error profiles of Oxford nanopore and Hepa sequencing. So I think we can add this analysis in that paper as well. Very good questions. Well, here's the I show you the performance of the error correction with respect to the coverage. Well, you will see that the more data, the more short we covering positions, the better accuracy you will have finally, right? So, and you have more than 8 reads covering a position, the error rate comes down to about 2% or even less than that. Well, and here's I show you an example how we use the error corrected long way to find the gene isoform. Okay, because actually the final goal is to find the gene isoform. This is the raw data, the raw reads. When we align to the genomes, well, we see a lot of insertion deletion mismatch here. But after error correction, this is the error correction. Well, after error correction, we have a much better alignment results. And also we rescue the alignment of 2 3'N exons. The raw data, well, in the raw data, because of the high error rate in these regions, they are not alignable, so you will miss these 2 exons. But now we can find it after error correction. Okay, this is an example of error correction. and also it's the gene isoform identification. But how we do the gene isoform identification in a genome world? So this is what we have, IDP, isoform detection and prediction. We have two parts, detection and prediction. I will show you why we have two steps of the ITP. Okay, well, to identify a gene isoform is equivalent to identify exon combination. For example, if you know isoform 1 contains exon 1, 2, 4, 5, then you identify isoform 1, right? So this is the same problems. However, if you use the exon as a structural units of isoform, well, then you will fail to find the novel events, for example, novel exon or novel isoform. Well, if you use the sample-specific exon identification results, well, then the result would be not very accurate because, well, they likely to use the coverage to identify the boundary is not very reliable. Well, so we have to change the strategy a bit. Well, instead of using the exon as the structural unit, we use the junction. Okay. In the same example, assumes that you know the 5' end, you know the 3' end. What you need to do is to find the combination of junction. Okay. Well, then you can identify isoform 1 by finding junction 1, 2, 3 together. Well, the good news is that if you have the short-read RNA-seq data, it's very easy to identify the junction, and the detection of junction is very sensitive as well. So they are reliable and also sensitive. So we use the junction combination for the isoform identification. Well, but how we find the junction combination? Well, now long we come back, okay? We have a long way here, then we align to the genome. Then we remind you that the long way is the cDNA data. So when you align to it, you have the fragment aligning to different position. Well, and you know there's a big gap, and this is likely to be the junction. If this junction detection from the long way is also supported by the short way, again, so we use short way, long way, short way. So we mix up them together. If this is supported by the long way, so this combination will be recorded. And when we have the combination of the junction, we compare to the existing, the annotated gene isoform. This step is called detection because we can detect it directly by this junction combination. If they match perfectly to an annotated gene isoform, we call this isoform detected. And when we check the detection rate versus the gene length, we find, well, again, this is old data, but the new one gives you about 90% of the short genes now. Well, but in the original publication, we find that the very short genes only give you about 80% detection rate. Okay? For the very long one, you miss a lot. The identification is about just 30%. Well, it makes sense because, well, this is the annotated long gene isoform. Well, the peptide sequencing data only captures a small fragment of it. You still miss this part and this part. So we have to find a way to rescue this very long gene transcript. Well, this is why we have the second step, prediction. This is why ITP4. Well, we have 3 steps in the prediction. At first, we construct the isoform candidate, and then we estimate the gene isoform abundance, and we select the isoform by the relative abundance within a gene. Well, the first step, isoform candidate reconstruction. Well, this is the long way you have, and this is the junction detected from the short way. Again, short way, long way, short way, right? Well, and we know the 5' end and the 3' end, then what you need to do is check all the combination of junction 1, 2, and 7. Because junction 3, 4, 5, 6 has been linked together by this long way, okay? So you do not need to check the combination of 3, 4, 5, 6. You just need to check the free junction here, 1, 2, 7. You have the 3 free junctions, so you have a combination, and then you just check, okay, which one would be true. Well, actually, the long way data is kind of similar to the penalty is in some existing statistical model. Well, so that we can shrink the search phase much smaller. Originally, if you only have the short way, you have 7 junctions. The combination of 7 junctions would be 128. And now, because of the long way, we just need to check A isoform candidates. Well, it's much easier to find the true one from A candidates than 128 candidates, right? So, but how we select from these A candidates? Well, this is isoform abundance estimation model developed by Jones and Wang in 2009. well, we can, this is a number of remapping to the different exon, and the parameters we're going to estimate will be the abundance of each isoform. And it assumes that the numbers of re follows a Poisson distribution, and they're going to solve the log likelihood of this Poisson distribution. Well, and when you take log of this Poisson distribution, you will find that this log likelihood will be a concave function. Then you can just apply any typical convex optimization algorithm to find the solution to solve this MLE. Then you have the estimate of the theta I. Well, when you have the theta estimation, the abundance estimation, then we pick up the relatively highly expressed isoform because, well, we have this isoform abundance from the short read coverage because it depends on the numbers of short read mapped to the exonic region. And the isoform candidate are generated from the long reads, right? We have the backbones from the long read, and you use the short read junctions for the isoform candidate construction as well. So if you have a high abundance assigned to the, one isoform, it's likely that you have a better match between two experimental data, the long read and short read. So we pick up the highly expressed one after the abundance estimation. Then we use it as a classifier parameter to check the the sensitivity when we control the false positive rate. When we control the false positive rate to about 5%, IDP gives you the sensitivity about 60%, but coupling gives you about 22%. And when we relax the false positive rate to about 10%, well, we have about 72, and coupling have about 44. So when you have a very reliable isoform reconstruction results, well, coupling gives you a very poor performance. And we applied our methods to human embryonic stem cell H1 cell line. This is a very famous cell line. Well, we find a numbers of isoform, and about 111 discoveries are the novel genes. Okay, although H1 has been studied for many years and by many publications, but we still be able to find a lot of novel things. And when we compare the expression of these novel genes in H1s, comparing to 16 human body map data in different tissues, we find that most of them have almost no expression level, and H1 have high expression level. And when we zoom, okay, this 1, 2, 3 is just the index of the novel gene. I pick up the top 10, okay? The first 8 have almost no expressions in 16 human tissues, but only high in the H1. When we zoom in the number 8 novel gene, we find almost nothing in REP-seq, Ensemble, gene codes, and UCSC annotation library. But we find very complicated genes in this region. And also we find number 8, I'm sorry, you may have questions that, well, you can find this novel gene in H1 because they have high expression in H1s. So that you can find it, but if you check the number 9 and number 10, you find that they also have the high expression in the other tissues, but nobody can find those before. I will address these problems in the coming slides, why we can find those, but not the other RNA-seq data can find them. Well, in particular, we pick up the top 24 novel genes and study their functions in the polypotence regulation. We find 3 of them called HPAT2, HPAT3, and PHD3. This is HPAT5, have a strong interaction in the gene regulatory networks of the polypotency marker nano, SOC2, and OCT4. Well, and here we'll address your concerns why we can find it, but no peer-reviewed study can find it. We check the sequence, and we find that about 70% of the sequence of these novel genes are repetitive. Only a small proportion of the novel genes are uniquely mappable. So imagine that you have these genes, and this part is repetitive. You have short read aligning to here, then you will ignore it because they are multiply mappable. And you have a small part here. And if you have long read, can capture this region and also can capture the uniquely mappable region. The whole thing, the whole reads, the peptide read will be uniquely mappable because you can capture a small proportion of the uniquely mappable region. Then we find that, then we're very comfortable to say that there's something there. This is how we find these novel genes. And well, and we have this paper published in Nature Genetics for more functional analysis of this novel gene. If you are interested, you can go through it. Well, and this study has been highlighted by Nature as well. Okay, we identify isoform and we quantify the isoform from the hybrid-6 data. And this is another tool we developed, IDP-ASC for allele-specific expression. Well, I think you are all familiar with this biology, but I'll just go through it briefly. But we have two copies of genes in the human genomes, right? So there's paternal and maternal ones. And after transcription, you have a different expression of these two alleles. Right? Saying, well, the paternal alleles express 4 molecules, the maternal express 2 molecules. This imbalance is called allele-specific expression. So we need to quantify this imbalance of expression, and we also need to solve a problem's haplotype. Okay? Previously, we only know here's a SNP AG and AC, TG, and AC, but we do not know the combination of this SNP in paternal alleles and maternal allele. This is so-called a haplotyping problem. We need to solve this too. Well, if you only have the short way, it's not likely to detect multiple SNP in one go. Yeah, because, excuse me, we have, well, the average distance between 2 SNPs is about 600 base pair. They're too far away. A single short way is about 200 base pair, 300 base pair. So a single short way cannot capture more than once. So, but the high accuracy of the short way can guarantee you call the SNP at a single site with high accuracy. Well, but if you only know here's AG, here's AC, TG, and CA here, and then you make the random guess, the successful way will be pretty low. When you have 2 SNPs, we only have half successful way. When you have 5 SNPs in your genes, well, you only have about 6%. It's pretty low if you just do the random guess. Unfortunately, in human genomes, we have significant numbers of genes containing more than 1 SNP. Okay, you have to consider the haplotyping problem. Now, long way come back. Well, long way is much longer than short way can capture multiple SNP in one go. For example, here, this long way can detect all 4 SNP in a single read. But we also have to consider, well, here's an error. We should only have a G here, but it gives you T because we have a high error rate, 13% error rate in the PEP-R sequencing data. And the sequencing bias, low throughput, site selection processing, PEP-R sequencing will all use also a big problem when you want to build up a statistical model for quantification. Okay, so we have to consider this problem so that why we have the hybrid again, we use the accuracy of the short way to guarantee the SNP calling of each side. Then we use the linkage information from the long way for the haplotyping. Also, the high throughput data from the third generation sequencing platform will allow us to do the quantification as well. Okay, this is how we do that. At first, we need to define where we want to study, how many SNP you're going to do. But we use the short way to call SNPs, okay? Saying you short way, you can call here as 4. Okay, M equal to 4 here. We have 4 SNPs in your gene. And we define haplotype this way. Well, for the J side, let W0J and W1J be the 2 SNPs you call from here, okay? I'm sorry, the 2 alleles from single SNP here, A or G. And let H as unordered pairs of haplotypes, H equal to J equal to 0 if it fits to the reference, otherwise it gives you 1 if it fits to the alternative. This is how we define the haplotype. And now we define the data. Okay, this is how we define what we want to solve, and this is define how we have on hand now. Well, suppose you have n reads, okay? And let S be n by m matrix, and the ith rows of this matrix correspond to the ith read, okay? It's very straightforward. This is how you have this matrix. And let Xij equal to 0 is matched to the reference alleles at the position I, position J's, and let it assign 1 if it matched to the alternative. If 2, it is the other 2 nucleotide will assign 2 or 3. If it is I's we cannot cover a position, we will assign a hyphen here. And then we compare, we check how similar these given haplotypes and your data. And if they match, and then we give 0. If they match to the H bar J, then we will give 1. If it's just an error, then we just assign 2 or 3. Okay? And this Xij will have a categorical distribution. I will show you later. This categorical distribution can be calculated from an error model input by the user. And actually, we can calculate this from the sequencing quality score as well. I will show you later. Well, now we have H, haplotype H, and also the data. We also denote row from 0 to 1 as the gene level ASE. Okay? And what we need to do is to fit a model of our data to find the most likely haplotype and the rows. And we know that row is unknown and is continuous. It's impossible to enumerate all the possibility, okay? Well, instead, we can draw the samples from the joint distribution of row and H. Well, here's we adapt a Bayesian framework when we have a non-informative prior. So it's very simple to have the model as likelihood of your data. Suppose all these are conditional independence given the haplotype H and the error model Qs. Well, we can simply have the products over the likelihood of all these here. Well, this is something has been developed a couple years ago. The people, a group in San Diego developed this model. However, this is for the DNA data. When we consider the RNA data, we have to account for the allele-specific expression. Well, now each V, xi, can be modeled as a mixture distribution with the way, row, and 1 minus row. I remind you that this is the gene level allele-specific expression. Well, and we will have these two items here, okay? And furthermore, we will calculate this F and this one. Well, assume each variance cause in each V are independent, conditional on the haplotypes and Qs and the rows, then, well, if the variance is not covered by a read, then it contribute nothing to the likelihood, so that we will have this. And each variance here, we have a categorical distribution with the parameter Pij. And this Pij can be calculated from the sequencing score. Well, for the base it called from the data, we'll give 1 minus the error probability. This error probability is encoded by this quality score here, Eij. And for the others, the remaining 3 nucleotides, we just split the error probability evenly. And this is the way how we calculate the likelihood. That means you can draw the sample from this likelihood, I mean the posterior distribution. Well, we use the slice sampling for rows and the typical Metropolis Hastings samplers for the haplotype. And we run 1,500 iteration with the first 1,000 iteration as the burning. If the conversion is not mapped, then we will run another 500 iteration at a time until the conversion is mapped. Then you have the MCMC sample, and then you can find the most likely H or row here. Okay? Well, then we apply to the GM12878 cell line. We know the haplotype of this cell line so that we can use this as the gold standard to evaluate the performance of IDP-ASE. Come back to this figure. Well, we know that if you make the random guess, this is the successful way you will have. So we do not expect any worse performance lower than this curve, right? Well, we apply our tools with the high-Brexit data or the short-read only data, and also compare performance to the M-base, which is an existing allele-specific expression methods. Well, we find that, well, the red colors, the high-Brexit data, can give you much better accuracy than the blue color here. So that means the high-Brexit data can give you much better prediction in terms of the haplotyping. Well, but only if you have a good tools to run the high-Brexit data, you will have this benefit. Otherwise, if you check the dash lines, the red dash line and the blue dash line, that means the M-base using the high-Brexit data or the short-read only data, there's almost no difference. Yeah, because M-base only use the marginal allele accounts of the rematrix. Remember, we have the rematrix just now. Well, if they only use the marginal numbers, they will lose the linkage information. So M-base cannot make use of the long-read information. So that's why you almost have nothing improve from this M-base results. And we also check that, well, if we have a better performance when we have more long read, yeah, definitely, well, use the high-Brexit and IDP-ASE, we have a better accuracy when we have more long read. And when M-base use, you cannot find much difference because they cannot make use of the long read information. Well, in terms of the quantification performance, we show that we compare the results using the long read data, the green color, or the short read data, the blue color, and also the high-Brexit data. We find that the long read only data give you the worst. Okay, this is the error. Well, give you the highest error. And the second best is the short read data only. As I mentioned, long read is not very good for quantitative analysis, but short read is good. And when we have the high-Brexit data, we have the best. estimation because you have a better haplotyping results, and you also have a short way for quantification as well. And when we check our distributions of the results, and we have a better fit to the simulations, but M-base cannot have a good performance in the middle, say 0.5. That means when two alleles are very similar expressions. And here's I show you the isoform, the second step of the IDP-ASE. Well, originally in this isoform abundance model, we have three isoforms. We want to estimate theta 1, 2, 3. And now we have a bit more complicated model. We have the maternal isoform and paternal isoform. Now what we need to estimate is 6 parameters here. Well, assumes that you can estimate theta k0 and theta k1, then you can calculate the imbalance, the allele-specific expression at the isoform level, tau k, by calculating their proportion like this. Okay? Well, but how do we calculate the isoform abundance of the allele-specific isoform? Well, we make a small modification of the Poisson model I mentioned previously. Well, we assume the numbers of Rhe falling to the allele-specific regions follow a Poisson distribution with this parameter. What is this alpha? Alpha Skj is an indicator that, well, whether it's a Rhe generated from the S regions, the K isoform, or the H haplotype. And we use 4 indicator here to represent the numbers, the Rhe's in different regions, because we have 2 isoform and you have 2 alleles, right? So this 2 by 2, you have 4 index. The first half is for the haplotype H, and the second half for the alternative haplotype H bar. And the first column is the index in the first column is for the first isoform, and the second column is for the second isoform. The third is for the first isoform, and the last column is for the second isoform. Take this as an example. Okay, we have 3 Rhe's. Yeah, say 3 because they are generated from the paternal haplotype. So that's why you have 1, 1 in the first half, and they can generate it from both isoforms. That's why you have 1, 1 in both columns. And in this example, we have 1 Rhe from this region because could be generated from both isoform. Is generated from the H bar here. So that's how we calculate the numbers of Rhe, and this number of Rhe, y-axis will follow this Poisson distribution. Then you can solve this by the same likelihoods and the convex optimization problems. Okay, then we solve the allele-specific expression now. I show you something very interesting. Well, in these genes, we find 2 SNPs here. Okay, well, SNP 1 have 42T and 84C. And the second SNP have 24G and 17A. So if this is the only information you have, then let's make a guess what's the haplotype. I think it makes sense that saying C and G together and T and A together, right? Because G and C contribute the most, the higher coverage in each position. Okay, this is so-called the pseudo-phasing, if you only consider the number of Rhe's. However, the gold standard tells you T, G, and CA is the true. Why? So anybody make a guess why this is the truth, but we have this coverage? Yeah. Why they have different expression? Yes, exactly. If we have used the IDP, we find there's 2 novel isoforms of this gene, and these 2 novel isoforms only express SNP 1. This skips SNP 2. So that's why you will have this coverage, but this is the truth. And actually, the long we tell you, the long we can detect TG and CA as well. So this is the beauties of long we. Because we have the long we can have a better haplotyping, and we have a more reliable isoform library so that you can have more reliable allele-specific expression analysis like this. Okay, we show you some different type of the allele-specific expression at the gene level and isoform level. In this one, we have 2 isoforms of these genes, and they express some exclusive SNPs. Okay, they have exclusive expressions. And in this one, they have some exclusive SNPs expression, and also they share the SNP 1 between these 2 isoforms. And then last, this is more interesting. We have 3 SNPs, 3 isoforms. And 3 isoforms express all SNPs, okay? There's no isoform-specific SNPs here. But we can tell the difference between them. Tau here is the imbalance of the expressions in each isoform. We find that the first isoform almost have 0.55 here, that means there's no preference between the paternals and maternals. And in the second one, we find the tau is 0.63. Well, it prefers to express the reference alleles, and this one, 0.33, well, it prefers to express the alternative alleles. Well, why we can find this? Because NIP1 located in the flanking region of the junction. And the junction here are the isoform specific junction. That's why we can make use of this junction allele specific way to find this different imbalance. Of course, when you want to calculate this, well, we have the biostatistical model like what we built just now. Okay, I show you some very interesting results from IDPSE. At last, I want to show you what we find from the fusion gene. Well, I think fusion gene, are you familiar with fusion genes? Okay, well, fusion genes are like this. Well, we have gene A from chromosome 9, gene B from chromosome 22. Well, in the cancer, well, cancer samples, well, they can be translocated together like this. Two chromosomes fused together, and you will have a fusion gene. After transcription, you could have something like fusion transcript. Well, this is very important in cancer because they are likely to be units in the cancer sample. Well, they can become a drug target as well. I don't want to go through all the possible regulation raised by the fusion events, but they are very important. We want to study the fusion events so that we can, and we have to solve 3 problems step by step. At first, you have to annotate the fusion events by giving 2 gene names, right? Which pairs of genes fuse together. And secondly, you need to give the position of the fusion site, where they fuse together. And at last, you need to tell what is the transcription product if you have the transcription events from this fusion genes locus. So these 3 things we need to solve. Well, if you only have short within 50 base pair, and this 25 and 25 base pair brings the fusion site, and then you align these 3 back to the genome, this is what you have because they are too short. 25 base pair, 25 base pair, you have a lot of possible hits. Okay? And then when you check the possible combinations of these hits, well, then you have too many pairs of this combination. And the multiple alignment and also the misalignment will bring you a lot of false positive. Well, so the existing 2, we have a very problematic output of this and this. And very few can output the fusion transcript identification. This is the problem of the existing 2 based on the short width. But if you have long width, you have 2,000 base pair long width. And this fragment's 800 base pair, this 1,200 base pair, is likely to make them unitarily back to the genome like this. Okay, you only have 1 pair. It's very simple. Yeah, this gene and this gene fuse together. You do not consider any other possible combination. So we can solve this problem very simply by just align the long width to the genome. And how about the second part? Well, fusion site. Well, we know that when we align the long way back to the genome, the high error rates of the PEPAL data is likely, well, it's very difficult to find the fusion site because of the high error rate of the data. The boundary of the alignment end here is not likely to the fusion site, okay? But we know the fusion site is not far away. It's in the neighborhood, okay? Well, then we can cast the flanking region around this fusion site of, say, 2,000 base pair on the left-hand side and the right-hand side. In total, we have 4,000 base pair and merge them together like an artificial reference sequence. In this artificial reference sequence, we have both fusion site contains, right? And then you just align the short way, like the RNA-seq alignment process, then you can find, because this is the genomic sequence, the human genomic sequence, so we do not have any error rate here, and the short way is high accuracy as well. So when you align the short way to this genomic region, you can find the single nucleotide resolution results. Now we solve the second problem, but how about the last one, the isoform? Well, we talk isoform for the whole seminar. Well, we have IDP, right? Well, I do not go through the technical details here, but we have to do some modification so that we can consider the fusion gene as a single gene, okay? And then we can apply the IDP so that we can identify the IDP, the isoform expressions and their abundance. And in order to evaluate the performance, the accuracy of the fusion gene detection, we generate a gold standard containing 7 fusion genes from 7 papers that are all validated by single sequencing or the PCR. So we know they are true. And let's see how many gold standard we can detect from IDP and the other short read methods. Okay, this is the short read methods. we have very good sensitivity. We find 24 gold standard. And the other tools, top fusion find 16, Sophie's 10. Well, we have much better sensitivity. Well, except diffuse. Diffuse gives us 27. It's higher than ours results in terms of sensitivity. However, in total, it output about 196 hits to contain 27 gold standard. If you check the gold standard rate, it's just about 14% of gold standard rate. Okay, so we have a high sensitivity, and we have a very good precision in terms of the gene fusion detection of this gold standard. Well, you may ask me a question. Well, if we have more data from the short read, whether we have a better fusion gene detection. Well, it sounds makes sense, right? Because the more data, the more information you have, the more reliable result you will have. Yes, when you require more short read support in this these tools, you will find the gold standard rate go up. When we require 10, well, I think the gold standard rates go up to about half, 50% now. But even you require this 10 short read, it's still a significant gap between our methods and their methods. Yeah, because we have long read, and long read give you some very reliable, very valuable and exclusive information short read cannot give you, even you increase the number of short reads. Okay, well, in terms of the fusion site determination, I show you an examples of AIB1 and the novel gene in chromosome 1. 6 long we find this novel fusion events, okay? When you align, part of it align to AIB1, part of it align to chromosome 1. However, the alignment end of the 6 long read here locates in a large region, 37 base pair region. They are not consistent. And in the right-hand side is 65 base pair region. So they are not consistent. You do not know which nucleotide position is the fusion site. However, when you align 31 short read to this region, you will find they all align to this position. Okay, so this is the fusion site, and this validated by PCR as well. And I want to share you some interesting things we find from the isoform expression. In this one, well, we only have one fusion site. The fusion site is also the fusion splicing event because this is RNA-seq data. You have a single nucleotide, single fusion site, and the original, the alternative splicing event in this gene, we produce 2 isoforms. And in this one, we have multiple fusion splice events, and the original alternative splicing event in the BCAS3 can also give you the high diversities of the isoform expression. And here, well, we have multiple splicing event between 2 genes, and also multiple alternative splicing event in both genes as well, and give you 3 isoforms. It's very complicated isoform expression from the fusion gene. And we also find some novel things, novel exon here. But this fusion event has been studied previously, but nobody find this novel exon. This is 2 novel exons, which is not annotated by any existing annotation library. But the long read alignment always find this novel exon expressed in the fusion events. And in this one, AIB1 and the novel gene in chromosome 1. AIB1 is a famous gene in the breast cancer research because the high expression of AIB1 can have a high effects to the tamoxifen treatments. And we find AIB1 have a novel exon here, and also it fused with 7 exon genes in the chromosome 1, and no annotation information reported by RAPSE, Ensemble, or the other annotation library. Okay. Well, now come to the last slide we have. Well, not really the last, but almost. Well, we develop a pipeline for transcriptome analysis using the short read and long read. The first step is for error correction, so that we have a much better quality long reads here, and then we use both for isoform identification and quantification. Then we do the haplotypings and also quantification at the gene level and also the isoform level of the allele-specific expression by these 2 IDP-ASE. For the fusion gene, we have IDP fusion to determine the very reliable fusion gene detection, and also determine the fusion site at the single nucleotide resolution, and also identify and quantify the isoform expression from the fusion genes. And actually,